potential damage would be if we didn't protect from solar radiation. And this allows us to kind of pick and choose the areas to implement this overhead irrigation methodology. A lot of our growers are saying, hey, we'll just irrigate everything because we don't want to lose any fruit. And that is also an option. So this is the approach. We have our little drone, we fly and we sample different parts of the bog and are able to come up with an estimate of the fruit exposed on that bog surface. So that allows us to do some site-specific planning and decide when and where we're going to use this evaporative cooling. So that is actually my last slide, or this is my last slide. And I just wanted to emphasize that our problems here in New Jersey are that we sort of walk this tightrope. And we're trying to walk the tightrope between overheating and over irrigation. And I think the really important thing to remember with irrigating to prevent overheating is short. Keep it short. You don't need a lot. Irrigate the top inch or two of the canopy, and that's where you're going to get all the evaporative cooling from anyway. Allison, I hope I didn't go over my time.
We're just by like 3 minutes, so we're in pretty decent shape. I think we'll go ahead and ask the two questions that came in via the chat, and then we'll, while Didier gets his screen ready to share. Question one: is there any relation between scald and soil tensiometer readings or soil moisture?
So no, not that I know of.
Cool. The second question is: I know scald is due to heat. Is that because at a certain temperature the fungi begin to quorum sense, or is it just due to being physically cooked?
Wow, I love that question. That's great. No, it's more of a cooking phenomenon. Okay. The fruit are when they're red and they can't evapotranspire, they'll literally just get hotter and hotter. Imagine, for example, if you were unable to sweat, how hot it would seem, especially those hot, dry California days.
All right. But good answer to a good question. I think we will go ahead and, Didier, if you want to unmute yourself and share your screen, we can dive into biological controls and then the fireworm timing. Once Didier is done, I will give some updates on the pesticide applicator training, and then we'll go to Q&A for everybody.
Well, first off, thanks for the invitation. And I was glad to hear that you guys started talking more and more about biological control in Wisconsin. So I'll present here some sort of summary from my own master's, some of my PhD. So I'm still a PhD student, still haven't finished that yet, but on biological control using Trichogramma. And the focus here is really, can we upscale things up? And you'll see why in just a sec. So just a quick summary on the black-headed fireworm. It is a major pest in cranberries, especially in organic production. And really, it is the worst for us in organic. So the first picture you saw on my presentation board is actually the favorite picture of my colleague Francois, because it shows what only one spray application missed. What happened there is that one part of the bloom actually broke down and didn't receive any insecticide. So a few days later, the bed was actually kind of burned, which is like why we call it black-headed fireworm. So, and this kind of thing, we see that every year. So, and just to emphasize again, on the left, this is berries that we collected in the fall. So when doing our damages assessment. So on the left, we've got a field with about 70% berries that are affected and gave about 40 barrels an acre. And on the right, 3% affected, about 180 barrels, which is the average in organic. But as you can see, it gives about $8,000 an acre on yield loss. So this is really a huge problem, and these are not isolated events. We see that every year. It's not always at the same place or the same bug, but this is very common to see nowadays in Quebec in organic production. So what's wrong? What happens is that, well, in the biopesticide world, well, first off, there is a very limited number of biopesticides. In Canada, we only have 2 spinosad and pyrethrins that are registered, but also there are limited options for potentially new AI. It's really like finding a needle in a haystack, because there aren't just many AIs that could work against these pests and that could be registered in organic. There's also a very, very long process of registration, especially in Canada. I don't know if we have some Canadians in the audience, but it is very a nightmare to get a new product registered. So this is also a problem, because even if we find a new product that works, we want to have it for many years. And there's also a constant threat of registration removal or new MRLs that will force, handlers to put some additional constraints. And also there's a general, public kind of a need and want to reduce pesticide. So this is sometimes attributed to the fact that there's a misinformation surrounding pesticides, but still we have to, navigate in that world where the general public wants less pesticide. So one of the solutions is biocontrol, which is the use of an organism to reduce population or density from another organism. And the typical biocontrol agent that we see are lady beetles, but here we'll talk about Trichogramma. And these are, minute egg parasitoids. They're about 1 millimeter long. And what they do is that, as you can see on the left, the female will lay her egg on the pest egg. So the larvae will develop just eating up the eggs of the pest, and then the pupae will darken the eggs. That's how we know that the egg has been parasitized, and then new adults will emerge. And by doing so, just prevent the pest egg from hatching. So there's been quite a lot of work in cranberries and Trichogramma over the past 30 years. And here I just summarized every species ever found in every regions that I know of, at least. But the real problem, and that's why I was talking about upscaling things up, is that the most common method to use Trichogramma right now is these little cards that growers have to go out and put in the field. And it really is like not doable for something as big as a cranberry bog. So, I mean, even if Trichogramma work, there's no way that anybody could use that with these little cards. So that's why we set up to develop a mechanized method for Trichogramma release and assess if it works. So specifically, we assessed two mechanized method, which is spraying and spreading. I'll come back to those later. And those have kind of two implications, which is the application itself and the incubation once it is applied. And it's kind of funny, my presentation's following Peter's, because I've talked to him about solar radiation and stuff like that when I heard his talk. I think it was last year, because that's also an issue for Trichogramma. The second objective is assessing, the optimal rate of application. The third, which wasn't an objective at first, but then became, because we thought at first that Trichogramma minutum from studies we did before was the best option for the black-headed fireworm, at least in Quebec. But due to some contamination, we ended up applying Trichogramma pretiosum in 2020, and that species performed actually quite well. So right now we're kind of in the middle of trying to find which the better one, or maybe it's a mix of the two. So I'll come back to that again later. And which generation of black-headed fireworm is most susceptible to it. So in terms of spraying, sorry for the mistake, and spreading, we have the first part, which is the application itself. So what happens to the eggs while they're being applied? So when we spray them, we just mix them with water and then apply them to the field. When we're spreading them, we mix them with moist vermiculite and put it in a fertilizer spreader. So for the spraying, the method is a bit more complicated because the usual pump of a sprayer, which is a centrifugal pump, breaks down the Trichogramma pupae. So we ended up creating this new prototype with a diaphragm pump and a direct injection. So what it does is that we're just spraying water, and then we're injecting directly after the pump some solution with the Trichogramma. And like I said, for the spreading, we're just mixing it with vermiculite and applying just like if it was a fertilizer. So what it does is that in the end, we found out that spraying, which is in blue here, does not affect the Trichogramma emergence, while the spreading does affect it and decreases it about by half, so which is pretty significant. And the reason behind that is pretty much the collision that happens in the spreader. So imagine if you are a Trichogramma egg in the tube of the spreader, and then get pushed down towards the end, colliding many times with the tube. In the end, you'll end up with a headache. So that's pretty much what happens to the Trichogramma itself. While in the spraying method, it's kind of protected by the water column. So it moves slower, and also in that kind of water protection bubble, which prevents any emergence problem. So in terms of application, spraying seems to be the best option, and spreading decreases by half. But the second part of the application is the incubation. So once it's applied on the bed, it has to sit there a few days before hatching. So what happens with this spraying and the spreading is that when you spray, droplets fall on the leaves, and the eggs will stay on top of the leaves. While when you spread, they'll fall off to the ground, and consequently, the eggs will be on the ground as well. And this is where Peter's presentation kind of mixed with mine, is that there's also solar radiation for the Trichogramma eggs, and their temperature will increase just like the berries. So this might cause a problem. And in fact, on the left, you can see the difference in emergence for Trichogramma being reared in the lab, so no effect of solar radiation. So as you can see, after only 48 hours, so if they haven't hatched in the first 48 hours, the hatching rate will decrease about the half. So that's where that's what you can see here. Well, if they're on the ground, no matter how long it takes before they hatch, well, there's no difference in emergence rate. So this is kind of the opposite that we had in the last trial is that if you spray, you lose about 50%, and if you're spreading, you don't lose anything from the 50% that were still alive after being spread. So then we went doing some, large-scale releases with those two methods. So what we did is in 2019, we used the Trichogramma minutum with 2 rates and the 2 methods, and we targeted the first generation. In 2020, we wanted to use minutum as well, but there was some mix-up and in the rearing process, and we ended up applying Trichogramma pretiosum with the same rates, the same 2 methods, and then we targeted the second generation. And in 2021, we did the 2 species together, still the 2 rates, but only 1 method of application, which was spraying, which seemed to be the best, even if we had some decrease after 48 hours. We tried to shorten the period before hatching of the eggs, and we did that for the first generation. So with all those trials, what we were able to compare is that which is the best application method, in the end, which is the best species, and overall, which is the best generation to target. So what we did is first we applied some sentinel eggs, which are the Mediterranean flower moth. So that's just a way to make sure we have eggs that they can parasitize if we don't find enough black-headed fireworm eggs. And then we search for black-headed fireworm eggs as well. We assess damages and yields at the end of the season. So here we have the results for the sentinel eggs. As you can see, there was no difference in 2019. There was a difference in 2020 between spraying and spreading. And this one is kind of a mystery to us still, but there were no effect of the application rate. And in 2021, we didn't see any differences in the Trichogramma minutum or pretiosum. Again, no effect of the application rate. What's the most interesting is the black-headed fireworm parasitism rate. So what we saw in 2019 and 2020 is no difference in between pulverization, which is spraying, and spreading. But there was an increase in parasitism, which is good. And I wanted to track here the scale of the effect. In 2020, we had Trichogramma pretiosum, and in 2019, we had Trichogramma minutum. And again, no effect of the application rate, but we had 55% parasitation, while we had about 18 in 2019. So we thought maybe pretiosum is better, but remember this was against the first generation and this was against the second generation. So then in 2021 we came back, we tested both species against the same generation, and then we saw there was no difference in between species. So the effect was probably caused by the generation and not the species. And this kind of fits earlier trials that were done in BC, and where they found out, even in the lab, they were using Trichogramma sibiricum back then, but even in the lab, parasitation rates were always better against the second generation than the first generation. So this kind of fits these other trials. In terms of damages, we didn't see anything either in 2019, 2020. We could not assess because, second generation eggs are overwintering eggs, so there's no effect on damages. And then 2021, no effect as well. This is kind of deceiving if you think this is a new method and that might help, but remember, we could not ask to any growers to just stop spraying with pyretrans or spinosad. So they still use insecticide. So that was kind of to be expected. Same with the yields, there was no effect for the same reason, pretty much. So in summary, what we saw is that spraying and spreading are pretty much the same, but they kind of induce a reduction of emergence by two different mechanisms, which is the application or the incubation. In terms of rate, there's no really difference in rate, so there's no point of going higher than 900 per hectares, just for cost-saving reasons. In terms of species, we don't know yet. We haven't seen any significant differences, but Trichogramma pretiosum tends to be a bit higher every time. So we'll have to, keep on investigating on that. And as for the generation, well, we saw that the second generation would be best. And I just wanted to end up by putting things into perspective. The best, case we had is 55% parasitism, which was with pretiosum against the second generation. And it might seem low, but you have to remember that insecticides effect is on only a portion of the whole population. So if you assess the efficacy of an insecticide and you get 60%, which is about what we get with spinosad, well, you can hit 60% of a certain part of the generation. So maybe 10%, and in second application, if you're in the highest of the curve, you can hit 60% of the generation, and then maybe 20%. So after, let's say, those three potential applications, you end up with a 54% efficacy on the whole generation, even though at each application you have a higher efficacy rate. And as for Trichogramma, when we assess the parasitism rate, we assess it for the whole generation. So the 55% is about the same in 3 application of spinosad. And in the end, 3 applications of spinosad and Trichogramma are about the same cost. So we have something that is potentially useful and comparable to organic insecticide, at least. So what's next? So we decided to pursue with spraying because it was better in terms of application. But what we can do is try to better synchronize the timing of emergence after application. And we can do that by bringing Trichogramma to the point they're really about to emerge, but keeping them in the dark, because they do emerge when the sun sets. So we can bring them in the dark to the very far point, and then apply them and make sure they will emerge within the first 24 hours. And what we want to do is to, bring it to a bit more commercialized way, because we were doing spraying with a prototype, and we started doing that with the Raven Sidekick system, which is a direct injector that is commercialized. But what we saw is that our prototype did not cause any reduction, but the Raven did. And the reason for that is probably the pump that is used. It is not a diaphragm pump, so we think the very key is the diaphragm pump. So we have been working to develop our own automated direct injection system. So we've been trying it in 2022, and we'll be for the next couple of seasons, but hopefully we'll have, a commercial product that can spray safely to Trichogramma.
I'm gonna go ahead and speak up. We're going through this program and then the fireworm timing as well. And if maybe we can put the fireworm timing on the shorter end, maybe 5 minutes or so.
Yeah, and the other part is quick.
Okay, perfect. That works out great then.
So that was about it for biological control. And then, so this is another project we've been working for the last 2 seasons. is to develop, an updated model, because our model in Canada did not work well for the black-headed fireworm again, and make it interactive as well. And you'll see how interactive can be to optimize spray application. So we're still working with the black-headed fireworm, but in the end, we want to, bring it to other pests at some point. But we started out with the black-headed fireworm because it's the worst for us. So what we see is that even if growers, scout, use threshold, and they use, best practices guide. This is a short guide we published last year. Even if they optimize their spraying equipment, we have this certification in Quebec that states that they have to be up to date, well calibrated, and stuff like that. Even if they use the same recommendation, and this is based by the fact they have this we have this club called CTAC, which is a crop consultant club. So 75% of our growers uses them for recommendation. So even if they all do their best, sometimes they have very good results and sometimes they have very bad results. And this is because even here we put the general curve of development between the eggs and the nymphs. We have 5 instars, so it only needs a shift in terms of phenology for the whole population being mixed up. And for example, here we have scouting 3 times. Every time they had the interventional threshold, so they sprayed 3 times. But on the green grower back down, they had fewer first instar and more last instar. And efficacy of pesticide isn't the same on L1 to L5. So it's always more effective on smaller larvae. And that makes the difference between a lot of damages or not at all. But also in conventional, that can make the difference between 1 spray or 2 spray needed. And the key factors in that is that, well, first, the grass isn't always greener on the other side, but it can be greener before. So the geography kind of is different. For example, in Wisconsin, if you take the phenology in the north and in the south, it's colder up north, so it will come up to the same point a bit later. And then the second point is that the size does matter sometimes. Like I said, smaller instars are more affected by insecticides. So the 2 key factors are timing and size. So what we can do is that we can compute that phenology using growing degree days, and target different periods, so hatching rate and stuff like that in time. So that's not very revolutionary. You guys have all sorts of tools to do that. Same here in Quebec, but these tools uses usually governmental weather station. So, and they compute what happens in between them. So this is the main growing region in Quebec, and these are like the 5 stations that we have. So all the growers within them are kind of assessed and not directly measured. What we have is ARTO tools, and most of our growers do have them now. So we can do sampling of the temperature directly at the farm level, which changes the definition that we have. So, and like Peter said, the temperature is taken in the bed and not at 32 feet like governmental weather station. So this is really where we have our insects. So I'll skip that pretty fast, but like I said, size does matter for insecticide efficacy. So what we did is first we took phenology on egg hatch pheromone trap, and what we did to increase the definition again is that we used automated pheromone trap. So we have a daily capture instead of a weekly capture count. We use data during mating as well. So what we did is to see what is the effect of rain, wind, temperature on the fact of if the adults mate or not. Because if they didn't mate because it was too windy, well, they might mate the following day or the other day, and then that changes when they will lay eggs. So that affects as well our model, not only with degrees, but with other factors. And then we used some data that was out there as well. Some of the data you have in Wisconsin, but all of the stages didn't match very well with our population, so we had to do some more sampling. So that was to update our model, and then we did efficacy trial of most insecticide that we have, and depending of the mode of action, so either contact or residual or both. So we did that for most insecticides that are used right now, conventional organic. So that was to make it interactive. So that's what we did. That's how it looked. So we developed that model. And the real difference is that emergence of G1 larvae, that was the comparison with our old model. So that's where the most of the different is. And we did set up this this platform, and what I will do, I'll just leave the presentation and show you the platform. So what we have is that every farm can access its own data. I have access to all, but they have access to their own. And what we can see is that we can go back, because now we're late, but if we go back in, for example, in May, we can see, well, up here, the egg hatch, down there, the adults, and down there, in the middle, all of the stages. So we can know at any given time, well, I have 10% that are in L2, 50% in L1, and 50% still haven't, 40% I haven't hatched. And this is based on their local data. So it's not assessed using, using other weather stations. It's really the weather in the field. What we can do as well using our efficacy data is to try some insecticide at different times. So for example, if I do a Spinoza there, that will be the effect theoretical on the population. But if I were to do a Spinoza later on larger instar, for example, doesn't work as well. And we have some weather effect as well. For example, there was a rain right there. So if I did a Spinoza right there, it would be just runoffs and nothing happens. So that's kind of a model that growers can play with to see when is the optimal timing. And also once an application is made, they can register it, and it changes, the curve shape for the whole season afterwards. So that's pretty much the tool that we developed. And this is still very theoretical, but
is that available for us to play around with, or is that still in development levels?
It's still in development. Even our growers don't have access to it, all of them. What we want to do, that was just my final point, is that next year we want to do some trial runs with pipe growers, a couple of crop consultants to see if that matches and if that really helps or it doesn't. I mean, we can develop a theoretical tool, but if it doesn't really help in the decision making, that's not worth anything. But we want to see, do you refer to it and does it help in general? So that's pretty much the tool. But yeah, at some point we want to extend it to other well, all of our growers, and maybe at some point further, just for growers to use that.
Thank you.
So that's pretty much it.
Yeah, that looked very cool. I have one question in the chat, and I will ask, what plant defenses have you seen, or what defenses does the cranberry have against pest pressures? If you have an answer on along those lines.
Well, we haven't measured plant defenses yet, but that's something we want to look into in the next couple of years, because we expect some, but we don't know yet what they do and how that does that translate. But we can expect some changes, you know, in physiology, but also some plant volatiles that they can emit. I know Cesar has been working on that for the last couple of years in New Jersey, so that's something that could be interesting. And
a question specifically on the trichogramma: are the eggs equally sensitive at all stages of development? Presumably that means to the pump, if you were seeing pump damage in the Raven sprayer, but go ahead and clarify for me, Peter, if I misunderstood.
Yeah, I don't know if it's for the pump or, you know, the solar radiation, you mean, Peter?
Okay, that could be too.
Yeah, either one. I was just curious.
Okay.
Like, I don't even know if there's different stages of development of the eggs. You know, I'm not an entomologist.
So typically when we apply them, they're in pupae within the egg of an alternate host that we use to just mass produce them. So we wait until they're in pupae. So they're all at the same stage when we apply them. So we didn't test if they were larvae or adults, but we can expect that adults would die in the pump. So that's very the stage where they are the most the most tough, because they're in pupae, and typically they spend about 6 days in that stage. So that's our window to apply them.
Thanks.
Awesome. Any other questions? Otherwise, yeah, let's just go ahead and do any questions for our presenters, either one. I want to thank the Growers Association, obviously, for partnering and presenting the brown bags with UW Extension. Didier Labarre from the Quebec Growers Association, Director of Research there, and then Peter Outemans at the Rutgers Ag Research Station. Thank you guys both for presenting for us and keeping us up to date on what research is going on throughout the world, so we can collaborate all the better. Otherwise, I'll wish you a very, a very Merry Thanksgiving and a, best of luck in the woods this week. Tom, if you want to give us a closeout as well.
I would just echo what you said, Allison, that on behalf of everybody at the Growers Association, we wish all of you a very happy Thanksgiving and hope you have a very good weekend with your family. And if you're going out in the woods, be careful and stay warm. And other than that, thanks for joining us, and we'll see you again soon on another brown bag session or another meeting on down the road.
Yes, indeed. Thanks. And Francois says good weekend. So, of course, happy Thanksgiving to all of the Americans, and the Canadians had their Thanksgiving a month ago. So continue being thankful. There's no harm. All right.
Thank you.
Thanks, everyone.
Bye.